Hi everyone, my name is Cristian Eduardo Gaitan Mendez, and I'm going to talk about this paper, and it's called Application of Fetusca rudinacea in Fitorremediation of Soils Contaminated with Lead, Nickel, Cadmium, and Petroleum Hydrocarbons. Well, first of all, we are going to talk about or introduce Fetusca rudinacea, and so we're going to start. Fetusca rudinacea is an species of grass, also known as tall fescue, and well, in the United States they also called as weed because they don't want to grow this kind of grass. But in this paper they use it because it has a lot of characteristics that makes a little bit different uh, um, from other plants, like it has heat tolerance, and it is important because where they made these experiments were in a place who has a lot of heat, and but more important, um, we noticed that this plant has a deep root system compared to another cool season grasses. So what they want to is to get all the contaminants that are in soil. So um, uh, so this is the reason why they use this plant and no other. Well, first of all, what is the phytoremediation? The contamination in soil may dissolve cell membranes of all the microorganisms that are in soil, and this is a big problem because uh, what we need is to get a lot of microorganisms in soil that makes easier the degradation of contaminants. So, well, and this is one of the problems that we have to solve or trying to solve with the phytoremediation. So, well, first, next, uh, also, when we have a lot of contamination, it may disrupt the plant root structure. So this is a big problem because most of the species in of plants that are in the soil are made well. It could generate aerotrophization. So that's a big problem here. So we don't want to. Well, on the other hand, we know that um, the fetusca, it's a plant that can get the contaminant by phytoextraction. So this is important because also, as we mentioned, uh, we need to get a plant that has uh, a big structure of roots. It would help to get all the contaminants into the plant. Well, in order to analyze these soils, they took three different kind of soils at different concentration of contaminants. As we see, we know that we could see in a red frame that they have the soil A. The first or the first frame is with the raw soil. It means that it doesn't have any treatment, and then the other has fertilized, and at the end with the triangle, um, we know that these soils were treated by with fetusca. So as we see in the first graph, we notice that the first soil for soil A, the we get the less concentration of contaminants with the soil that were treated with Fetusca. Also in the three from the leaf, we have the same results for the three contaminant soils. In nickel, we notice almost the same. We notice that in the soil treated with Fetusca, we reduces almost 100 um, mass of contaminant in the soil. So we notice in almost every graph that we get with soil A, we get the best results when we treat this soil with Fetusca. As we notice, we can say that cadmium was the most degradation. Well, this is another graph that shows us the degradation in the three different soils, and the raw soil, treated with fertilizer, and the third bar shows us the soil that were treated with Fetusca and fertilizer. So what we notice here, we notice here that hydrocarbons of petroleum were degraded, and we show it with the carbons, the number of carbons that it has. In the first one, we notice that where carbons degraded in a limit of C6 carbons to 12 carbons. And we notice that almost for the first soil, the number of carbons that were degraded most were those that has from 25 to 30 carbons, and also in the other ones. Also, they make an analysis to the mortality and inhibition growth. What we see here is that they make different experiments where they change the pH from almost these three different soils. Well, in conclusions, they perform experiment under pot conditions in the Fitorremediation assisted with fertilized treatments using the Fetusca rhodinacea plant on soils contaminated with occurring pollens like I mentioned, provide satisfactory effects. Fetusca rhodinacea were accumulated mainly in root tissues in following descent order, as we saw on first graphs. So this is all my explanation. I hope you were enjoyed, and see you.